So am I audible now? Can I start it this one?
Yes.
Yeah. So these are the two ways to do the selection. Either it can be the positive selection or it can be the negative selection of the cells. Then if you want to select the cells, so this is how it looks. So this is one of the experiment from the professor's lab. So he has done through the three things, whether it can be southern blot, northern blot, or the western blot. Do you people know what it is? What is southern blot? What is northern blot? What is western blot?
Sir, in chromatography,
Hmm? It is. Hello. What it is?
sir,
What is southern?
DNA.
Yes. What is northern blot? So southern blot, northern blot, western blot is to detect. Southern blot is to detect DNA, and northern blot is to detect RNA, and whereas western blot is to detect the protein. Okay. So this is one of the gene they wanted to eliminate from the genome. So if you see this plus region means we are diploid organism. So any this is a mouse experiment if I'm not wrong. So here also if you see it's a diploid. So in the both the strand they have to eliminate. So if they eliminated in only one strand, then they got this kind of results. And if they eliminated in two strands, then they got only one band here, which means they cut it, they have eliminated some region of here. And if in terms of the northern blot, if the wild type they have the RNA here where it is got mutated, or if it is eliminated, then it is not there. The same way in the protein level, in the wild type it is there, or in the double knockout, or in the not double knockout, it's in the both the stand if it is eliminated, then it is not there. Okay. So how they usually generate? Once those cells, whatever they selected from the homologous recombination, then they will again give it to the mouse blastocysts. It's nothing but the foster mother, which is hormonally very well versed, which can be made pregnant using this methodology. Then finally, the chimeric mouse will be there. So this chimeric mouse again with a pure mouse, it will be in breed will be happen. Then finally, the heterozygous pups will be there, which is having this one strand will not be there, and then the other strand will be uh, knocked out. So finally, they will do again the inbreed of this heterozygous, that is plus or minus. And we have studied through genetics. We know that mental experiment, if the plus or minus is there, then plus plus will be the first option, and then the plus minus will be the second option, and plus minus will be the second option, then minus minus. So which means in the 1 is to 2 is to 1 ratio, we will be getting out of 4 mice, whatever we get, one would be the two knockout, like both the strands of the DNA will be, or both strands of the genome will be the mutated one. Okay. So this is still now we know that how it is happening the whole cell. Like I don't want to mutate the whole cell, but if I wanted to mutate only one specific cell, for example the liver cell or brain cell, okay, then we would be doing the next system called Cree lot speed system, we say. That is Cree means Cree recombinase, and then the LOXP is nothing but a DNA sequence. Okay, so the Cree-LOXP system, how it works? If the both the LOXP site is nothing but it's a 34 base per region of the DNA. So once it has this kind of construct, or as HSVTK and then the Neo and then this one, if it is there, what will happen? Through the Cree-LOXP system, if the both the LOXP sites are in the same direction, then the gene will be knocked out. If the both the reload species are in the different directions, then it will be inversed and everything. So that we will be seeing within the next thing. So here you see, if the Kirillot speed system is there, the log speed system is there, then the Cree enzyme if comes inactive, then the this region will be deleted. Okay. So what will happen? That is, this is what I have been talking about. Log speed system is nothing but it is a DNA sequence. So wherever this kind of lock-speed system is there, then this Cre recombinase enzyme will comes and bind, which is around 38 kilodalton enzyme. Then it will be removing the gene of interest. So finally, the inactivated gene will be there. But if you want to inverse the region, okay, if this Cre system is in the opposite direction, then you can inverse the protein. Or if the locks are there in this kind of system, then you can do the translocation. So this will come here and then this will come here. So these kind of different system that we can do using the Cre-Loxp system. So the Cre-Loxp system overall, if you see, which is a Cre recombinase that creates the recombination, that's why it's called Cre recombinase enzyme, which is isolated from the P1 bacteriophage. Okay. So this is what I have been telling. Once you get the mouse, which is having the LOXP system with neomycin gene, then you will be mating with the Cree containing the mouse. Then once the breed is happening, then we would be getting the our animal of interest, which will be having the selection, like selected deletion. Okay, so this is what that overall thing that he has talked about. So far we have completed with the recap. Do people have any doubt, or else then we can go back to the question and answers? Do you have any doubt?
No, sir.
So shall we go to the question and answer? Okay, first question: which of the following statement is or correct? Hematopoietic stem cells are example of multipotent stem cells. Germline cell lines stem cells are that only produce sperms are example of totipotent cells. And third is transgenic mouse models can be used to investigate the immune defense against bacterial infection. So which are the statements are correct? First one is correct or wrong? Hematopoietic stem cells are example of multipotent stem cells. Is it correct?
Wrong, sir.
Why? Why it is wrong? Okay, the second thing, germline stem cells that only produce sperms are example of totipotent cells. Is it correct? Okay, so the answer first one is correct because hematopoietic stem cells will give certain set of cells, right? So that is correct. And then germline stem cells
Okay, sir.
that only produce sperms, that is not correct because zygote also will be there, egg also will be there. So that's why it is not an example of totipotent stem cells. And transgenic mouse models can be used to investigate the immune defense against bacterial infection. Yeah, that is also correct, which means option C is correct. 1 and 3 are correct. Okay. And next, which of the following statements is or correct? Lymph nodes are connected to the blood vessels and lymphatic vessels. Is it correct statement? Hmm?
Yes, sir.
Okay. Lymph nodes acts as a filters for antibodies. Is it correct?
Yes.
Okay. And B cells are present in the cortex region of the lymph nodes. That is also correct.
I am confused here.
Okay. So where it is present, B cells. So lymph nodes not acts as a filters for antibodies, right? What it does, it will make sure that it will act as a filter for antigen, okay, not antibody. Lymph nodes are connected to the blood vessels and lymphatic vessels, that is correct. Okay. B cells are rich in cortex region of the lymph nodes, correct? We told na there are three structure: cortex, paracortex, and medulla. So cortex region is rich in B cells, then paracortex is rich in T cells. Okay. So lymph nodes acts as a filter for antigen, not antibodies. Okay. So the option C
Okay,
is
sir.
correct. So 1 and 3 are correct. Understood? Okay, next.
Yes, sir.
Lamina propria is associated with this which one? Spleen with white pulp, thymic cortex, galt, balt, lymph nodes. Lamina propria. Where we have seen this word? Lamina propria. Okay, it came under pace patch. So pace patch is related to which one?
Mal.
Yes. So that is almost gut associated, right? It is in the intestinal, right? So intestine is gut. So mucosal immunity only, but it is specifically under galt. So gut associated lymphoid tissue. Okay, it is coming under Payer's patch. Okay, thymic stromal lymphopoietin or TSLP is produced by, this we have not seen, so I will answer. It's in the epithelial typho reticular cells. Okay, that we have not seen it. Okay, efferent lymphatic vessels in lymph nodes. That we have seen. Which one is the option? Is rich in FDCs and DCs. Is rich in antibodies and effector T cells. Is rich in germinal centers, and it is rich in antigen, or it is rich in hematopoietic stem cells.
Sir, B option have
Which one?
B.
B, yes. So efferent lymphatic vessels in lymph node is rich in antibodies and effector T cells. Okay. M cells plays an important role in which one? Phagocytosis, transcytosis, apoptosis, pinocytosis, and ferroptosis. Which one?
Phagocytosis.
Phagocytosis, M cells.
B, sir.
Which option is correct? We have seen the M cells. I will show you
B,
the.
sir. B.
What is
B,
transcytosis
correct.
then? This is a transcytosis. So what is the meaning of
Transcytosis.
transcytosis? What is the meaning of it?
I don't.
Transcytosis is nothing but, I told you an example. Na? If you are having the river, okay, you want to cross from one side to the another side, right? So you will be using the boat, right? So the transcytosis is a boat mechanism. What it does, it will take the materials from one side, then it will deliver to the other side. Understood?
Yes, sir.
Yeah. So if the region is there, like this is your gut, then the gut valve is there, then it the another place. So what transcytosis does, whatever the pathogen it is here, then it will be trapped, then it will be given the inside of the region. Okay. That is why it is called transcytosis. Okay. Then next question. Yeah. So here the option is transcytosis. The mice where we delete disrupt the gene of interest from a particular organ or type of cell is known as, if we delete from the particular gene of interest, it is known as knock-in mice, conventional knockout mice, conditional knockout mice, wild type mice, or GFP knock-in mice. If we delete only one gene from one, what is the meaning? Any guess? Or should I answer?
C, how, sir?
Yes, answer C. It's a conditional knockout. Okay. For successfully created knockout cells using homologous recombination. So where we have seen the first knockout, now which one is incorrect they are asking. Okay, they are not asking which one is correct. So the cells will survive on G418 containing medium. Is it correct statement? G418. Is it correct or wrong? So this is the correct statement. Okay, because neomycin will remove the G418. Okay, so second one, HSVTK is not incorporated, but neomycin is incorporated into the genome. So that is also correct. Cells will not survive on ganciclovir or acyclovir is wrong. Okay, cells will survive on acyclovir and then ganciclovir is correct, and cells will pass both positive and then negative selection. Okay, that is also wrong. Okay. To prevent the differentiation of stem cells, what is added into the medium that I have not talked about? So what they will do, they will use the leukemia inhibiting factor that is called LIF. So that they will use to prevent the differentiation of stem cells. Okay. Where do the clonal selection and clonal deletion of T cells takes place? Clonal selection. First I have talked about this topic. Which organ?
Sir, leukemia.
Huh?
Leukemia. Lympho nodes.
No. Clonal selection, clonal deletion. T cells. What is the meaning of T cells?
Thymus.
Yes, thymus. I told you, na, if it is in the same, if it detects T cell detects the our own antigen, then it cannot be survive. So it will go for cell death. So the clonal selection happens in thymus. Lymph nodes that we have talked about germinal centers and everything. We didn't talk about the clonal selection. Okay, understood?
Okay, sir.
Yes. So do you have any other doubts?
No, sir.
No doubt. What about others? If you have any other doubt, not only related to this lecture, if you want to ask other doubts also, you can ask. So that is what I have for the day. And who are answering that boy? I didn't hear your. Can you please introduce yourself? Someone was answering, na? One boy was answering, right? What's your name?
Take Raihan.
Okay. Where are you from, Raihan?
Kolkata.
Where?
Kolkata.
Kolkata. Okay. What are you doing? Like you are studying or?
BSc BMLT.
Oh, good. So for that you have taken this course. Good. So you have this paper immunology in your college.
Yes, sir.
Good. So what's your future plan next and everything? What do you want to do?
And job.
Okay. Great. So next week we'll see the same time, 7 to 8. So sorry, 7 to 9. We will be having one more session on week 3, and we will do. If you want to ask any other question, you can ask me. Otherwise, we can close the session for the day. Do you want to ask anything?
No, sir.
What about others? Chaitra, do you have any doubt?
No.
No. Monish, you can suggest me if you want me to improve something or something, then you can ask me to improve. Then I can come up with a new idea. Is it okay the way I'm going, or should I change anything in my teaching as well?
It is good.
Okay. So next week, shall we see then?
Yes, sir.
Yes. Okay. Okay, Monish, you didn't introduce yourself. Can you please introduce?
Sir, my name is Monish. I'm studying in Satyabama Institute of Science and Technology
Okay.
in Chennai.
Okay. So you're from Tamil Nadu.
Yes, sir.
Oh, so your native is Tamil Nadu.
Yes, sir. My native is Tamil Nadu.
Okay. What do you study?
BTech Biotechnology.
Great. I'm also from Tamil Nadu. I did my bachelor's in Coimbatore, and
In which school, sir? Which college?
PSG College of Arts and Science.
Okay, sir.
Yeah, then I joined in the University of Hyderabad for the integrated PhD course, and I did my masters here, and I'm continuing for my PhD. I'm in my
Sir,
third year
and
of
what are you
PhD.
doing PhD?
Yes, so I'm basically working on HIV TB co-infection.
Okay, sir.
So I am trying to understand what are the factors of MTB responsible for the HIV enhancement, because people have seen already if you have both HIV and TB in our body, what will happen? It will enhance each other. The viral titers will be more high. The people will leads to AIDS stage very fast when compared with the HIV alone people. So I am trying to understand why that fast forward reaction is happening during the co-infection state.
Okay, sir.
Yes. So that project only we have submitted to the PMRF to get this fellowship.
Yes, sir. All the best, sir.
Yeah, thank you so much. All the best to you too. So you are in masters, right? You will be having projects next year, right?
No, sir. I'm doing BTech.
Oh, BTech. But still, you people have projects, right? Monish also.
Yes.
Yeah.
Yes,
So
Yes,
sir.
where
I
are
sir.
you?
have a project. I want to do a project
Okay.
next year.
Where do you want to
I'm
do?
first year now.
Okay.
Next year I want to do a project. I have no idea, sir, about what do you want to do.
Okay. So you are asking suggestion or?
Yes, sir. Please suggest some
Okay.
information, sir.
Yeah, yeah, yeah. So see, for your BTech project now, what I would suggest, you have to do your at the end of your fourth year, you have to do project, right? That is compulsory in your component,
Yes, sir.
so that
Yes,
they will
sir.
allot the faculty and everything. If I'm not wrong.
Yes, sir.
Okay. So
Sir, we
there are.
have projects for each sem.
Okay, great. So what I would suggest you like other not only that. So if you see today's budget, Government of India announced PMRF again. Okay, they are going to give 10,000 people for the next 5 years. So in different institutes. So please aim for those fellowship, because that is the highest paid fellowship in India, right? So you be getting around 80,000. As of now, it is 80,000 per month salary. So they said it will be enhanced. So I'm hoping that it would be enhanced up to 1 lakh. Okay, at least 10,000 means it will be coming around 90,000 per month salary. So for that you
If
need
we
your.
do a project, sir,
Yeah. No, no. To do
if
your
we,
PhD, what I'm trying to say, during your BTech now, you have to do multiple internships. Okay. Because internships will carry marks in this kind of fellowships. They would be giving you 10 marks for your internships.
Sir,
Ah,
where we want to ask for internship, sir?
okay. So internship, there are two ways you can approach Monish faculties. One is you can write a mail to them. If you write 100 mails to different faculties, then you would be getting one or two replies. That is the reality, I would say. Or else you can ask your faculty who is having the connections in some other institutes. So they can help you to get into their lab. This is first. Second one is Government of India, time to time they will announce the internships. Many IITs are there, right? So if you do you know Biotechnica?
Biotic.
yes,
Some
sir.
Yeah, so
things.
Biotechnica. So it's a website. It's a private organization. Okay, so they you go and subscribe their email thing. So daily you will get one email which institute is announcing any kind of vacancy or something. So some institutes include JNCSR is there in the Bangalore. Okay, they will announce the internship time. So during your summer vacation time is there, right? So the application will start in the month of March. Okay, so
Okay,
that
sir.
Okay,
time you
sir.
can
Sir,
apply.
but for
Mm.
first years, like they're not
Second year or
taking, or
third
they're
year
asking.
second year you can apply. So what I'm trying to say, if you want to apply next year, start from now onwards to work on that. Okay, if they are asking SOP,
Okay, sir.
then start writing the SOP, then you give it to your faculty, ask them to correct it once or twice or multiple times, you give it to multiple people to correct it.
Okay, sir.
So that would be better. And you need one or two recommendations from people. Then if you get it, then it would be really really grateful. There are certain internships, it is very good paid actually. Per month they would be giving around 5,000 per month for your internships. So you would be learning new things. Then on top of that, you would be getting paid as well.
Is that a
Okay, sir.
biotechnic is a website, sir?
Yes, 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 website.
Okay, sir.
Yeah.
Sir,
So,
for first years, they asked us to go through an industrial training, sir.
okay, where are you planning to go?
No, sir, like I have no idea which university offers this, and we need to only go through some biotechnology side.
Okay. So industry you can approach multiple areas. Na? There are n number of startup companies are there in Tamil Nadu. So you can write it to them and then go directly and then do it. See, if you say biotechnology, then Avin company is also a biotechnology based only, right? Dairy farm and everything they are doing.
Yes,
So,
sir.
so you can write it to them, then you can pay and then go. There are certain institutes you can pay and then go. Some institute they will not ask you to pay anything. So
Okay,
that
sir.
way you can do. Take a list of government authorized things. If you want to get into it, if you can travel, if you are not afraid of traveling, then write it to our university only. University of Hyderabad has this good bionest we say. Okay, bionest is a place where multiple companies are doing this their own project, that startup project. So you can write it to the company directly.
Okay, sir. So if we write to University of Hyderabad,
Not
we'll get the
University of
training.
Hyderabad, Bionest. Bionest is a
Um,
separate altogether in University of Hyderabad. The place is inside
okay,
the
sir.
University of Hyderabad. So that you can do.
Okay, sir.
Then Dr. Reddy's laboratory is there in Hyderabad. In our campus only, there is one institute, Dr. Reddy's Institute of Life Sciences.
Okay, sir.
We call it as drills. So you can write it to the faculty and then you can get it. When you are writing now, don't write it to any faculty. Show your interest, like how best you can get into that. Don't write it that since my childhood I want to become a researcher or something. That sometimes looks like an artificial thing. Okay, so yes, we don't know now, but since childhood, some most of us wanted to become a doctor, and we didn't succeed in the NEET exam. Then we joined in the research. Right, most of us are like that.
Me too, sir.
Yes,
Yes.
sir.
So don't lie like that. So what I would say, if you ask me if you how you should write it to me, you should read my papers, my publications. Then if you find some of the techniques are really really interesting, then you can quote that things that this paper, that article showed that these technical techniques are there. I don't know how to use this. So if I get a chance to explore in your lab, then it would be really great for me to get other fellowships in my career and everything. You can write it.
Okay, sir.
Yes, and you people are eligible to do PhD without MTech, right? Because you are BTech people.
Yes,
Yes,
sir.
sir.
So just focus on those kind of things, how to get into IITs or ISER and how can shape your life, you should see. And now onwards, you should start reading on this GATE exam. Okay.
Yes, sir.
Yeah. You read a bit into pieces, and then you will get a really good picture at the end of 3 years.
Okay, sir.
So be focused, and if you want my mail ID, you can take my mail ID, and whenever if you want to contact me, you can contact me at any point of time.
Okay, sir.
So my mail ID is siranjeevirangaraj@gmail.com. Should I put it in the chat box?
Yes, sir.
Rangaraj@gmail.com. Okay. And next week also we will, because our whole aim to conduct this live sessions to transfer our knowledge to you people, not only in a subject wise, in other cases, how if you want to get an idea about your project or if you want to get an idea about your entrance exam preparation. So for that only we NPTEL started this live sessions with PMRF.
Okay, sir.
Yeah. So do you have any other question? No, other people they don't have any question. Darshika, Divya, Nandini.
Sir,
Yeah, Monish.
I missed the first part of the section, sir. This full video will come in NPTEL group, sir.
Yes, yes, yes. It will come for sure. So, but what happened? The first part means which topic you started listening?
I came at 7:30 or
Yeah,
7.
yeah, it's fine. See, whatever I have taken the first part, na, that already there, what Manish sir took, okay?
Okay, sir.
So you can go back and then see those things. I will also upload these videos, you don't worry.
Okay, sir.
Yeah, so next week also we will be having 7 to 9 class. So if you want to attend, then you can come. If you are not able to attend, then again we will upload the video.
Yes, sir.
Yes. So other people, any doubts? Have any doubt, then you can ask in the chat box as well. And if you don't have any doubts, then we can meet next week. Okay. Okay. You people can leave. Then I will stay for some more time. Then I will leave.
Okay, sir.
Bye bye. See you next week.
Bye, sir.
If there is any website to know about the internships for government hospital for
Government.
biotech.
Yeah, yeah. That's what I'm saying. Na, this Biotechnica is there, right? Uh,
Yes, sir.
so they will send you mail actually. Wait.
That internship will be done for working from home or sir or
No, you have to go there
when
in
to.
person. In person. You are a computational person, or how you? What do you want to do? Like, I'm asking. Like, you want to get into computational thing only. I want to do the computational work. I don't want to do with lab, or what is your preference? I'm asking.
I want to go to lab and know to
Things.
get some knowledge, sir.
Yes, yes, yes,
Skill knowledge.
yes. So that you can do
But
it for sure.
okay, sir.
So the most of the previously during COVID time they kept like online internship. Now everything is offline. So you will go to their place, then you will do the work.
Yes, sir. Thank you, sir.
Multiple things will open, Monish. For example, IITs will open their internship soon within 1 or 2 weeks. But you are in first year, you said, na?
Yes, sir.
Maybe next year you are eligible to apply. Then other institutes like TAFR Institute is there, na? Tata Institute of Fundamental Research.
Yes, sir.
So they will also invite for this internships. So it's a
Okay.
student visiting program or something. So they would be paying more.
This
They'll be
IIT
invited.
Chennai as a research center, sir.
Yes, IIT Madras is a research institute.
Okay, sir. I
Yeah.
will try to some.
You can write it to the faculty directly and then go.
Okay, sir.
So they will reply. That's what I said. Now you have to read their papers, Monish, properly. Then you can write it to them.
Okay, sir.
It should not be just like that. If you write it now, they won't reply because they might be getting hundreds of emails, right? So your email should be unique.
Yes, sir.
Yes, only then we would be getting the things done. Yeah. So I got your mail already, actually.
Yes, sir. I
Yeah.
send hi.
Yes, 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 yes. It's okay. I will reply back. Yeah. Okay. We'll see you next week. Okay. And if you have any
Yes, sir.
doubt, you can mail me at any point. Okay, Manish.
Okay, sir.
Take care.
Thank you, sir.
Bye bye. Good night. Yeah, you people can leave now. Good night.